My name is Andy Sarles. I'm an assistant professor of mechanical engineering, and I'm also a UT alumnus. I grew up in West Virginia, studied engineering at UT, did my graduate degrees at Virginia Tech, and in 2011, I was lucky to come back to Knoxville and join UT as a faculty member. While you might think I've come full circle, I've actually only traversed along a single diagonal line. This lame joke is for all the cartographers in the audience. So the lab I started upon my return is called the Bio-Inspired Materials and Transduction Lab. And today my goal is to convince you that water droplets can teach us something about making new types of smart materials that are inspired by biology. So a smart material is a material that couples different forms of energy. Piezoelectric ceramics are one type of smart material that deforms when you apply electrical voltage, or when you squeeze them, they produce an electrical signal. Other types include shape memory alloy wires and electro-optical liquid crystals. Shape memory or smart materials are useful in many applications, including unmanned aerial vehicles, the autofocus feature in digital cameras, the shock absorbers on a Chevrolet Corvette, cardiac stents, and even sports equipment all use smart materials. But biology is adept at coupling multiple forms of energy. In fact, living organisms are what I would consider the ultimate smart material, where much of this smartness results from basic functions like sensing and energy conversion that occur in individual cells and single biomolecules within the cells. But what is a bio-inspired material? I would argue it's not a leopard print coat. But in fact, wearable materials can be bio-inspired, like the swimsuits worn at the Olympics that are inspired by the rough skin of sharks, which helps swimmers swim faster by reducing drag in the water. Thus, there's a distinction between mimicking aesthetics and translating biological function. Other examples of bioinspiration include self-cleaning or super-hydrophobic materials inspired by the roughness of the lotus leaf, the Mercedes Bionic, a car modeled after the yellow boxfish, and even new designs for reduced drag wind turbine blades modeled after the fins of humpback whales. My work is motivated by the many functions performed at the cell membrane or lipid bilayer, a selectively permeable structure composed of phospholipids and other biological molecules that regulate transport in cells. The question my work asks is, can we use artificial cell membranes to make new types of smart materials that use biomolecules? A phospholipid is a molecule that is amphiphilic, consisting of one portion that does not like to be around water and a head group that does like to be around water. This property allows phospholipids to self-assemble in the presence of water, and it's a property that we use to help them assemble into the proper structures for our biomolecular smart materials. I'll bet that you know more about self-assembly than you think. If you've ever blown bubbles, you've witnessed self-assembly. Soap molecules, which are also amphiphilic and which are similarly structured to phospholipids, self-assemble in the presence of water at the air-water interface to stabilize the shape of the bubble. Consider two scenarios. In one case, two water droplets in oil are brought into contact. The result? They coalesce to form one larger droplet. But if phospholipids are added, they spontaneously self-coat or self-assemble around the exterior of the water droplet such that when they're brought into contact, they do not coalesce. That interface is called a droplet interface bilayer. We form droplet interface bilayers by dispensing small water droplets, roughly 1 millimeter in diameter, into shallow wells filled with oil. And when they come into contact, they form this artificial cell membrane. One advantage of the DIB, or droplet interface bilayer technique, is that droplets can be arranged in any number of ways to construct single and multiple lipid bilayers. We can even make red droplet interface bilayers. Two droplets yields a single lipid bilayer, but more droplets produce many biomolecular interfaces. A challenge to this work is being able to accurately dispense and arrange each droplet in the network. But we discovered that if you put the water droplets in oil in a flexible, specially designed flexible cartridge, bilayer formation is as easy as squeeze and release. Here, we included transmembrane proteins that affect the transport across the bilayer, and these spikes show that ions are being transported across the membrane. Hopefully I've convinced you that playing with droplets can be used to form lipid bilayers that mimic the basic structure of a cell membrane. For the next few minutes, I want to discuss some potential applications for droplets and lipid bilayers that my group is currently investigating. Humans have roughly 16,000 cells in each ear devoted to hearing. These hair cells feature small hair-like protrusions called stereocilia, which bend and move as sound waves enter the ear, a process that triggers an electrical pulse being sent to the brain. We're working to develop synthetic sensors that also use membranes and synthetic hairs for sensing airflow and even sound as a potential replacement for hearing aids. A new project in our group is to develop biomolecular smart materials that sense, actuate, and convert energy, and which feature a large number of unit cells composed of biomolecules. My group's contribution to this work is to develop ways to efficiently fabricate these multicellular embedded systems. Lipid bilayers are often used as platforms for studying biomolecules that pass through or reside in a membrane. In this vein, we are studying the insertion behavior of botulinum neurotoxin into synthetic cell membranes. Commonly known as Botox, this neurotoxin is used for wrinkle reduction, but is also one of the deadliest neurotoxins known. In addition to bacterial toxins, we're also starting to investigate how a synthetic cell membrane can be used to listen to bacteria. We're working with Dr. Gladys Alexander in the BCMB department to use a lipid bilayer to intercept and analyze chemical packets called outer membrane vesicles, which bacteria emit for infecting hosts. Lastly, we're working with scientists at Oak Ridge to explore less invasive optical methods for probing biomolecular activity in membrane structures. Here, the goal is to quantify transport of species and information across these molecular barriers without the need for conductive probes. So thank you for your attention.